population genomics of the Asian Ubara bustard. So Ubara bustard is actually a generic term for species that belongs to the Chlamydotis genus. These are laking birds that belong to the Otididae family. For those that not maybe aware of it, laking birds are actually birds where the male is displaying to showcase its quality, and female are choosing the male in terms of sexual selection. The most known case of a species like that is a peacock that most people are aware of. And those birds are inhabiting steppes from the North Africa and the Middle East and Central Asia. And there's actually two species of Ubara. The first one is the North African Ubara Chlamydotis undulata, whose range is all the way towards North Africa. It contains two subspecies, one is endemic to the Canary Island, Chlamydotis undulata fuerteventurei, and another one, which is all around the North Africa, from the Atlantic coast of Morocco all the way to Egypt, which is Chlamydotis undulata. The second species is the Asian Ubara, Chlamydotis mcqueeney. Here you have a picture just to compare of two males, one of North African Ubara displaying and the Asian Ubara displaying. And this species has also very large range, started from the Sinai region to the Gulf region, Iran, Pakistan, all the way to Central Asia, all the way to Mongolia and China. And both species are actually facing very similar threats, one being the habitat degradation and loss of habitat. You can see here, and human encroachment. You can see it's a picture to illustrate. It's a Ubara that was found dead at the bottom of a power line. It's in the Fuerteventura Island. Here, Ubara that look a bit lost, roaming around in field crop. And also, one of the big issues is overgrazing. You can see here the potential impact of overgrazing. You have an area that's fenced, so there's no grazing, an area that's open to grazing. And you could imagine that for birds that living in steppe area, that can be that has a very significant change on their actual habitat. Another threat that you might be aware of is actually poaching and unregulated hunting. So there's hunting mainly in the Gulf, from people in the Gulf on falconry, but there's also people in Asia who's hunting Ubara with guns and even machine gun. And to go back to the topic also of today, there are potential threats from global warming. So this is not a Ubara, it's a related species that's a little bustard. It's a European species. And I'll present you the results of a work that was done by another team. And what they looked at is the impact of heat and temperature on displaying behavior of the male. And I was saying to you, it's a laking species, so the displaying behavior is a very crucial aspect of their social system. And what they can show here on the y-axis, you have the displaying intensity of male and the temperature here, and you clearly see a decrease of displaying intensity in relation with temperature. And that leads them to conclude that they anticipate a decrease in daytime display activity of up to 10% for the temperature increase projected by actual model of global warming, which is significant, 10%. And in the Ubara, actually, we have some clues that global warming might be affecting the species, but we need to delve deeper into it. So this is a graph to show the average temperature in eastern Morocco, where we work, over the last 20 years. And what you can see is that the mean temperature increased on average by 3 degrees, and the minimum temperature increased on average by 7 degrees, which is very significant. And what it translates to is that, in bird, it's been shown there's been different, there's a good corpus of publications showing that there's potential effect of climate change on the bird demographic, being direct or indirect. So that could be related to breeding, behavior, and survival. And in the Ubara, we have some clues. We have to work deeper to confirm whether it's related to it or not. But this is an illustration about it. So you have on the graph the productivity, the average female productivity, depending on the origin of birds, be either the birds captive bred or wild bred, and the age, so from 2, 3, 4, and 5 years old. And you could see here on the blue is the productivity of female over a period, an average productivity between 2002 and 2011. And in green, or dark here, you have the average productivity of female for the last 10 years. And you can see that there has been a very significant decrease of productivity over the last 10 years. So that could also affect very seriously the sustainability of the species in the wild. So both species are actually recorded as vulnerable today by the IUCN. And as you know, they are emblematic species for the falconry. And falconry, for those that's not in the Gulf, is a very important aspect in the Gulf, and actually been recognized by the UNESCO as a living human heritage. And Ubara is the main quarry for falconry. So because of that, the Abu Dhabi government decided to initiate some conservation program to both preserve the species in the wild and also sustain falconry. So sorry about that. So linked to this, they initiated and implemented different conservation breeding. One in Morocco is the Emirates Center for Wildlife Propagation, who's working on the North African Ubara. One in the UAE, so the National Avian Research Center and the Sheikh Khalifa Ubara Breeding Center in Suyan. It's about an hour away from Abu Dhabi. And one in Kazakhstan, the Sheikh Khalifa Ubara Breeding Center. And all those programs are actually managed by the International Fund for Ubara Conservation. So now I'm going to focus more about the Asian Ubara, the Makwini. Here you have a larger distribution of the species. And there is one specificity, is that we can find both forms, birds that are strictly resident and birds that are migratory. And here, the stripes, the orange stripes are indicating the resident population. And what we can see is that they're fairly scattered and they're isolated and also kind of a small size. And if we look now at the migratory population, you have in gray the breeding range of migratory population, and you can identify 3 migratory pathways. This one we identify with a very extensive radio tracking, more than 1,000 birds. And so we have the western flyway, where birds are going along the Caspian Sea, then moving towards the eastern Euphrates region all the way to Jordan. The central flyway, where birds are going south, some arriving in the Gulf, and some stopping in Iran and even in Pakistan. And then the eastern and far eastern flyway, which is very impressive in terms of migration, because you have birds leaving the Gobi deserts, going all the way to Kazakhstan, around the Himalaya, going back to Pakistan, and even some of them are stopping in India, in Rajasthan and Gujarat. There's been some work that was done before on the species in terms of analyzing genetic structure. The first work in 2004 was done using mitochondrial DNA. The second work that was done in 2012, published in 2012, was using microsatellite data. And the results of both those studies are very similar, that they identify limited genetic differentiation throughout the range, and mainly driven by isolation by distance. So that's why we wanted to work further and use some genomic data to look a bit deeper into this. The first stage was to create a reference genome for the species, because there was not none, and then use this to do resequencing data to identify conservation units within the species, and eventually proposing conservation action. So for the reference genome, we selected one bird, which is actually today a founder of and is still alive in the flock, which means that we're going to keep it as a reference. And that's a bird that was collected in the wild as an egg in 2005 in Yemen. And to do the genome, we generated long-read sequencing using ONT sequencing capacities. And we did the assembly annotation using our own pipeline. So the output of it, doesn't want to go, there you go, was we obtained a genome of about 1.2 gigabytes with close to 900 countings, but also L50 of 16, and a very good coverage. And also what's important for the following part of the study is 97% of the genome was contained in contigs that were at least 1 million base pair. So we use this to, after do our resequencing project. Here it's a map illustrating the sampling for the project. So we sampled 10 populations, 3 of resident population. So here in Israel, Yemen, and southern Iran. And 7 populations that are migratory birds. So northern Iran, West Kazakhstan, West Uzbekistan, Central Uzbekistan, Central Kazakhstan, Eastern Kazakhstan, and then Mongolia. And then also those birds, we've been able to validate that they're actually migrant birds. Here you have the track of the birds that were sampled. So you can see the one sampled in Caspian Sea were actually following that western flyway. The one from central Kazakhstan, central Uzbekistan were following the central flyway. And the one from Mongolia and eastern Kazakhstan were following the eastern flyway. So there we can confirm we have a fairly good representation of the different flyway of the species. So to summarize, we sampled 114 individuals for 10 species. All migratory pathways are represented. And it's the most comprehensive sample of the species to date. Actually, we sampled most of the remaining population. All these samples were analyzed using a short read from Illumina, and variant calling and filtering was conducted after in-house. Globally, in terms of free sequencing results, good. Yeah. We obtained an average of 2 million reads per individual, and an average coverage of 25x. We analyze them first to look at different genetic diversity levels, starting with heterozygosity. Then we also look at run of homozygosity as an expression of potential inbreeding level in the various population. And to analyze genetic structure, we did different analysis, but I will present here the results of our discriminant analysis of principal components and population admixture. So in terms of heterozygosity, here you have a graph that shows the heterozygosity for each population, and you can see that here you have the resident population, and globally they show a lower heterozygosity than the other population. And if we look at the population that are currently considered as a core of the remaining species, Central Uzbekistan, Central Kazakhstan, and Eastern Kazakhstan, see that the heterozygosity is slightly higher, and then it decreases also in Mongolia. So and when you look at the run of homozygosity and the number of run of homozygosity, we found also a pattern where resident population from Israel and Yemen have a fairly higher number of run of homozygosity compared to the rest of the population, where the numbers is fairly similar. And if we really want to look at some, trends, we can see that also the core population seems to exhibit lower amount of run of homozygosity. And there, if we look at the length of those runs, not only the numbers, but how big they are, you can see that Israel has the higher length in run of homozygosity. Then we have a group which is slightly lower, and the core population here, then it's also lower from the rest of the group, translating like less inbreeding level. In terms of population structure in DAPC, when we look at the whole data set, the first thing is that the DAPC is actually, we can see it's coherent with the geography, where we see a gradient on the axis, the first axis, going from west no, east to west. And 3 populations shows as distinct: the population in Yemen, here the population in Mongolia, and the population here in East Kazakhstan. And then if we look deeper at that big block or potato here, we can look, there is an apparent substructuring where Israel population is showing on its side. Then there is a second cluster where we have Iranian population with central population of Kazakhstan and Uzbekistan. And a third cluster here with population from western Kazakhstan and western Uzbekistan. And when we look at the admixture results, the first thing is that actually the results are coherent with the DAPC. So there's a convergence of the data, where we can see isolation here of Yemen, East Kazakhstan, and Mongolia. And then within the remaining population, we also see the substructuring with Israel showing a side, southern Iran, northern Iran, central Kazakhstan, Uzbekistan together, and western Kazakhstan, Uzbekistan on another fine. So the results are coherent between 2 analyses. And also what's happening is that with both this analyzing, we don't see a clear signal of the migration. We know the migration is the birds are going back to to where they're born, so it's very strict in terms of different pathways. There's a clear different pathway, but we don't find that signal in the migratory behavior. Oh, I'm going a bit fast, maybe. So in terms of conclusions, if we look at the genetic diversity, we can see that the heterozygosity decreased toward the range and the edge of the species. And, but more important, the Yemen population shows the lowest level of genetic diversity and the highest level of inbreeding. So that population might be in a need of genetic rescue. We also have some data in terms of demography where that shows that the population is not doing that well. And the next stage of what we're going to do, we have some historical samples from 70 years old and even some of them for a bit more than that. And what we want to look is what is their genetic diversity and run of homozygosity to put our results in perspective and see if what we see today in Israel, for example, and Yemen is a resultant of a long-term isolation or a recent decrease and collapse. In terms of population genomics, so we see mainly that the genetic differentiation is not driven by migration. And it might sound a bit contradictory, but the resident populations show higher level of genetic diversity between themselves and the rest of the range, and which is consistent with a hypothesis of reduced gene flow between sedentary and migratory population. So how does it translate in terms of identifying conservation units that will be unit or population that will need to be managed independently in terms of the conservation program? So the first thing when we do this, we are not only considering the genetics, but also the demography and the ecology of the species. So the first parameter is the fact that the resident population are scattered and isolated. We have limited genetic differentiation throughout the range with the marker used. Here we didn't look yet at adaptation. It's a next step. And what we see is that, to come back to the tracking and migration of birds, is that the tracking data of birds reveal a potentially very strong genetic component to migration. So the graph on the right is illustrating an experiment we conducted a few years ago where we did a common garden experiment. So if you recall, we have a breeding center located here in central Kazakhstan. And the founders of that center are representative of all 3 migration routes, some from West Kazakhstan, Central Kazakhstan, and Eastern Kazakhstan. So what we did is that we bred chicks in captivity from East and West Kazakhstan. And just to give you some indication, where our practices in terms of breeding in captivity is all artificial. We do artificial insemination, genetic management, artificial incubation, and rearing. So the birds cannot learn anything from female in terms of migration or anything. Then, so the birds produced here, I'm going to focus more on the birds from eastern Kazakhstan region that are in blue, where they're transferred to West Kazakhstan. There's no scale here, but it's a massive distance. And were released. And what we can see is that captive bred birds from one generation or two in captivity were able to migrate as their wild counterpart. So the genetic behavior, the behavior of migration was maintained in captivity. So they did properly. We can see here the red one, which are the birds originated from West Kazakhstan. Then they got lost and got stuck in the mountain here. And if we look on the other side of the country, in West Kazakhstan, birds in blue here of eastern origin, they kind of migrated like if they were in the eastern part. Instead of going along the Caspian Sea, they went through the Caspian Sea. Some landed up in Russia and very far in Russia, Turkey and everything. And one point that's important is that the birds are migrating alone. So they're not learning migration from their conspecific. So it means that here we have captive bred birds who express a genetic behavior. We don't know exactly what it's going to be, but so it's fairly important in terms of deciding how we're managing the population in the future. So because of that, in our conservation program, we manage each of the population independently. And we're going to do future work to investigate what are the drivers of genetics, the genetic driver of migration in the species. So what could be the impact of climate change? And actually the first two talks this morning gave me lots of ideas of things we can do. This is related to the previous slide. So we know that the migration is genetically driven, which is good in captivity because it means we can maintain it in captivity in terms of conservation. When we release the birds in the proper area, they migrate like they want. So for today, it's good. But what's going to happen when the climate will change and the birds are going to have to move or adapt, shift range, or maybe what if we do assisted colonization to area where the species actually got extinct? And this is actually an experiment we did last year to try to see how the birds are going to behave. So we took birds whose genetics is originated from eastern Kazakhstan. So it's about, and they were bred in captivity. And we released them in another area on the border between Russia and Mongolia, which is about 1.5, 2,000 kilometers further east. And when we look at their migration pattern, they actually got stuck here in the mountain. They did not really adapt to the environment, so they did what they were supposed to do should we have released them there, but they got stuck here. So their migration was not successful. So this can be a problem in future for the survival of the species, because yet we don't know how the species will adapt in terms of climate change, because maybe the timing required for adaptation is not going to be in the same magnitude, if you can say, with the timing of the current climate change. So. And so in order to work on this aspect and go deeper in terms of the migration of the species, we're now working on an improved genome of the species, where we moved from 900 contigs to 100, and the L50 from 16 to now 8, and with even more contiguity, and that will be good because that will help us to look at local adaptation and genetics of migration. And also, if we've managed to identify some clues as how the migration is driven species, and we can see if there's any way where we can help the species colonizing or recolonizing other areas and survive in the on the longer run. And yeah, so I'm on time. So I have time to acknowledge all the rest of my colleagues. You can see it's a large range, and I don't put data, but it's thousands of birds has been equipped with transmitters and everything. So it's a collective work. So I have to acknowledge the conservation breeding, my team, the conservation genetics, the ecology and fieldwork, the GIS that's giving us very nice maps, the research group, the veterinary, and also acknowledge the founder for the program. Yeah.